First of all, thank you for inviting me. It's a really great honor to have been invited. So as Leo mentioned, I did my PhD with Scott at the Stowers Institute in Kansas City. And I don't think I would have ended up here at UW in this program if it wasn't for Scott. So choosing the right mentor is definitely a good choice. And I've learned two things this morning. The first is, during graduate school, I always had the sense that Scott put way more confidence in me and my experiments than he should have. And it sometimes made me question his senility, but in retrospect, it worked out really well. And the second thing that I learned today was, so as a resident, I order a whole lot of EKGs. I probably ordered a couple hundred this year. About 5 of those were for chest pain and kids that actually kind of scared me. And the other couple hundred was to measure QT intervals. So I now know who to thank for that, and that's Barry. I'm an expert at reading EKGs now because of that. So what I want to talk about today is something work I started a couple years before graduate school, trying to understand how double-strand breaks are repaired. It was really fun to put this talk together because it gave me an opportunity to look back and it's fun to look back and look at the amount of work that you've covered in a period of time. I don't think people really take the time to appreciate that. So what I'm going to talk about is how inversions affect and alter double-strand break repair outcomes. Let's see. Let's use the computer. So as most of you know, double-strand breaks are made during prophase of meiosis 1. And obviously, meiosis is a specialized form of cell division in which a single diploid cell will become either 4 haploid cells or 1 haploid oocyte. And the cell will do this by going through 1 round of genome duplication followed by 2 rounds of cell division. And importantly, during meiosis, homologous chromosomes must pair, double-strand breaks are made, and then some of those double-strand breaks, but not all, are repaired as crossovers. And it's these crossovers that help to ensure the proper segregation of homologous chromosomes during the subsequent meiotic divisions. Now, in Drosophila, as in most organisms, more double-strand breaks are made than are actually repaired as crossovers. So the majority of breaks are actually repaired as gene conversions. And these are non-reciprocal exchange events that involve copying of sequence from one chromosome onto another. In Drosophila, conversions are quite small, maybe 500, 600 base pairs, although I've seen them as large as, say, 8 to 10 kb. But the question that we had is, what's the distribution of these events? Now, the distribution of crossovers, you might imagine the distribution is fairly even no matter where you look along a chromosome arm, with the frequency being the same. That's not actually what we see. What we see in reality is the majority of crossover events are distributed along a distal 2/3 of the chromosome arm. And that distribution is caused by the presence of the centromere itself, not this pericentric heterochromatin, and it's known as the centromere effect. Now, a second mechanism that affects the distribution of crossover events is called interference. And what interference is doing is that when 2 crossovers occur on the same chromosome, it's working to ensure that they're widely spaced. So not only is having a crossover on a chromosome essential to ensure proper segregation during meiosis, having 2 crossovers that are close together can actually cause missegregation in meiosis. That's true in flies, and that's true in humans as well. So in Drosophila, it's quite rare to recover chromatids with no crossover events. It's quite common to get homologs with 1 crossover event. And because of interference, it's actually less common than you would expect by chance to get homologs with 2 or more crossover events. And when you do recover those are typically quite widely spaced. And then finally, Barbara did a really nice job of this just now, but I do want to mention the karyotype of melanogaster. So melanogaster has 4 chromosomes, 2 large autosomes, an X chromosome, and then a small, mostly heterochromatic 4th chromosome, which does not undergo exchange. Each of these chromosomes have large blocks of pericentric heterochromatin around the centromere. And then the melanogaster females are generally XX and males are generally XY, and I'm not going to show the Y chromosome here. So our question was, could we identify outcomes of double-strand break repair genome-wide using genome sequencing? And besides being a really cool thing to do, because it's a lot of data and it's a lot of programming, and those are 2 fun things, it would let us answer 2 previously unclear questions about gene conversions in Drosophila. And the first one is, how are they distributed along the chromosome arm, genome-wide? And are they sensitive to the same controls, specifically the interference and the centromere effect, that crossovers are? So to do this experiment, what we did was we crossed 2 isogenic lines, Canton S and Y1118, to create heterozygous females. And these females were heterozygous for about 500,000 single nucleotide polymorphisms. We then whole genome sequenced 196 individual males from these females, and we used changes in polymorphisms in order to identify sites of crossing over and gene conversion. So what did we find? Well, I'm showing you individual events here with chromosomes along the x-axis and then a subset of individuals along the y-axis. And you can see from this data that we identified sites of single crossover events, we identified double crossover events, and we were also able to identify gene conversion events as well. So it's a lot of fun to stare at individual crossover and gene conversion events, which I did for a very long time. It's also quite interesting to actually consolidate them all into an aggregate view. And when you do that, so here I'm showing you all the crossover events on chromosome 3L. Single crossovers are on the top, double crossovers are on the bottom, or in this down here. And you can see that the majority of crossover events, as you would expect, are distributed along the distal 2/3 of the chromosome arm. And because of interference, the double crossovers that we recover are quite widely spaced. They're about 10 megabases apart. So our data from whole genome sequencing for crossovers matched what we would have expected using visible markers. But what we didn't know was what the distribution of gene conversions would be, and whether or not conversions would be sensitive to the centromere effect or interference. So this is because previous work on gene conversion was done at one gene in Drosophila for the most part, called Rosie. These were done using convert my way or die experiments in which rare gene conversion, the only individuals who survived were those that had rare gene conversion events within Rosie. And this was a lot of work. This involved setting up thousands of crosses in order to recover just a few progeny. This is more work than I wanted to do. I just wanted to program. So from our whole genome sequencing data, we were able to identify 291 gene conversion events in the 196 progeny that we sequenced. And all those events are plotted here for each chromosome. Now, when you compare these events to the crossover events, hopefully what you appreciate is gene conversions are much more evenly spaced along the chromosome arm than crossovers are. And indeed, if you look in the proximal 1/3 of the chromosome arm, for gene conversions, you see essentially the same number of conversions here as you do in each other third of the chromosome arm. So this is telling us that conversion events are not sensitive to the centromere effect. Now, if conversions aren't sensitive to the centromere effect, the next obvious question is, are they sensitive to interference? And when we look at individual gene conversion events, we find that they are not. So we were able to identify instances of gene conversions very close to one another. We found them very close to single crossovers, very close to double crossovers, and even within double crossovers. So these are observations that we obviously couldn't have made using visible marker-based approaches. The only way we could have identified these was by genome sequencing. So overall, what did we learn here? Well, we know that we can identify sites of crossing over in gene conversion using whole genome sequencing. We now know conversions are insensitive to the centromere effect and interference, unlike crossovers. And we also know that conversions are relatively evenly distributed along the chromosome arm. So how do we use this knowledge in order to address our question, which is how do inversions actually alter double-strand break repair? Well, in Drosophila, we have a really nice collection of chromosomes that are multiply inverted and have multiple inversions, and they're called balancer chromosomes. So we wanted to know how double-strand breaks are actually repaired on those balancer chromosomes. We had a problem. We didn't know the sequence or the structure of those chromosomes. So we set out to determine the sequence and structure of balancer chromosomes in Drosophila. So what is a balancer? I think most people in the room know, but I'll go through it really quickly. A balancer is a multiply inverted chromosome that not only prevents recombination, but prevents the recovery of recombinant products. Balancers carry dominant visible markers that makes them easy to track in a stock. And they generally have a recessive lethal or sterile allele that will prevent them from becoming homozygous in a stock. So how is it, in the rare instances in which there's a crossover between a balancer and its normal sequence homolog, how is it that it prevents exchange? I think Larry would have liked slides like this, and he probably drew out things like this a lot. So here I'm showing you a single inversion, a paracentric inversion. And you can imagine that any single crossover event within an inverted segment is going to lead to two products, one acentric and one dicentric chromosome. And neither of these are going to segregate properly at the first meiotic division. There's another type of inversion, and these are ones that involve the centromere. These are called pericentric inversions. Now, in single crossover events within pericentric inversions function a little bit differently. So any crossover is going to give you two products with centromeres, so they're going to segregate properly during the subsequent meiotic divisions. But they have large deletions and duplications that are going to be lethal. So as an example, the top product here has 2 copies of the A region and no copies of the D region. So there's multiple balancers available in Drosophila, but we focused on the most commonly used X, second, and third chromosome balancers that are shown here. So we used a combination of Illumina short-read sequencing and Nanopore long-read sequencing in order to identify all the breakpoints, determine their sequence, and then also study the diversity among these chromosomes. So the balancers we sequenced carried 44 total inversion breakpoints, and using our approach, we were able to precisely identify the location of all 44 of those inversion breakpoints. We validated the majority of them with Sanger sequencing, with PCR and Sanger sequencing, and then in those cases where the inversions had been previously reported, what we saw matched that previous report. So these 41 inversions directly affected 31 protein coding genes, most of whose function is unknown, but I do love to point out that the third chromosome balancer TM3, the 94D inversion, bisects all isoforms of the conserved tumor suppressor gene P53. And I like to point that out because people treat balancers like they're some type of wild-type background in crosses, when in fact they're actually highly mutagenized, rearranged chromosomes that wouldn't really last very long if we put them in a population. So careful with your balancers, people. So now we knew the structure, we could determine the sequence. And the reason we wanted to know the sequence is because we hypothesized that identically labeled balancer chromosomes would have subtle differences between them. So balancers were created one time, and then they were distributed among different stocks. So you can imagine over time that they're going to pick up mutations. And we were curious if we could see evidence of those mutations. So here I'm showing you data for the X chromosome balancer. So we sequenced 8 X chromosome stocks. They're here along the Y-axis. And then position along the X-axis, position on the X chromosome is the X-axis here. And then any green box is a unique polymorphism that's only present on that balancer from that stock. And hopefully what's obvious is there are 3 very large tracts of sequence diversity, of unique sequence, on these bottom 3 FM7 stocks. So what happened? Well, there's subtypes of these balancers, as many of you know, FM7A, FM7C, et cetera. And FM7C carries a female sterile allele of a gene called singed. And you can imagine that gene actually lies within the 4D to 11F inversion breakpoint, right in the middle. And you can imagine that any opportunity to lose that gene would quickly be taken over and would quickly be selected for in the stock because you've lost your female sterile. And that's exactly what happened here. So while single crossover events with balancers are lethal, a double crossover event is actually perfectly viable. So here, these 3 stocks had a double crossover event that replaced the mutant copy of singed with the wild type copy, and they subsequently took over the stock. But regardless of events like these, we're actually able to determine the sequence of all these balancers. Okay, so knowing the sequence of the balancers, we can now ask a more specific question about how frequently exchange events happen with balancer chromosomes. So we hypothesized that if we could find a balancer that had an unbalanced region, we would see evidence of historical exchange events in that interval. And we knew that the third chromosome balancer, TM3, had a large block of unbalanced sequence on the distal end. It had about 7 megabases. So we thought that if we sequenced a panel of TM3 chromosomes, we would see evidence for historical recombination events on these chromosomes. That's actually exactly what we found. So we sequenced 17 different TM3 chromosomes. And here I'm showing you a similar heat map where unique polymorphisms are plotted in green. And hopefully 2 things are apparent here. First, some of these crossover events are quite similar to one another, which reflects the shared ancestry of some of these chromosomes. But second, we can actually see for the first time the distance over which an inversion breakpoint is suppressing exchange. So this inversion is right here, but we don't see any crossover events within about 1 to 2 megabases of the inversion breakpoint. I know the data set is small. It's only 17 individuals. It's only 10 crossover events. But still, it's the first time we can see visually how that inversion breakpoint is suppressing repair of double-strand breaks as exchange events. Finally, we can come back to our original question, which is how are the breaks actually repaired on these balancer chromosomes? So there was evidence in the literature that double-strand breaks could be repaired as gene conversions with balancers and their normal sequence homologs. And what we wanted to find was evidence of historical repair events in stocks. So we looked at 4 of the 8 FM7 chromosomes that we sequenced, and using a highly stringent approach that under-identified historical gene conversion events, we identified 24 gene conversion events on these balancer chromosomes. And I'm showing you all of them here in green. So the point isn't the number, it's the position. So you see gene conversion events within every inverted segment. And you also see gene conversion events very close to inversion breakpoints. So this is telling us something very interesting, and it's that double-strand breaks are made essentially on top of inversion breakpoints or very close to inversion breakpoints. And the cell is left with a problem. It has to figure out how to repair those. And it can actually do that by gene conversion, but not by exchange. Okay, so back to the original question. How is it that inversions are altering double-strand break repair outcomes? So we know that we can identify conversion events and crossover events using whole genome sequencing. We know the structure of the balancer chromosomes, and we also know the sequence of the balancer chromosomes. How do we put all this together to answer our question? So it's been known for some time that heterozygosity for an inversion breakpoint on one chromosome arm will increase the frequency of exchange on those chromosomes that can undergo recombination. This is shown very nicely here by Sturtevant and Beadle back in 1936. They didn't need R, they could just draw graphs like that, which are really nice. And what they're doing is they're showing you the frequency of exchange on the y-axis along a chromosome arm, and in this case it's the second chromosome. And in the presence of a third chromosome balancer called pain, it's not a great balancer, I think it's 1, maybe 2 inversions, you see that the frequency of exchange in the centromere, proximal, and telomere regions goes up. That increase in exchange is more pronounced in the presence of a better balancer, CLB on the X chromosome, and it's even higher in the presence of a third and X chromosome balancer. So this phenomena, an increase in exchange on a freely recombining chromosome in the presence of an inverted chromosome, is called the interchromosomal effect. So despite being described nearly 80 years ago, the mechanism behind this increase in exchange is unknown. So is it that there's the same number of double-strand breaks, which are somehow switched from the gene conversion pathway to the crossover pathway, or are new double-strand breaks being made in response to these multiply inverted chromosomes? Second, the question of how those breaks on chromosomes which cannot undergo repair by crossing over, how they're repaired, is unknown. Is it gene conversion, like what we saw with our balancer chromosomes, or is there another mechanism here that we weren't aware of? So in order to test this, we set up 2 different crosses. First, we created females who are heterozygous for an X and third chromosome balancer. So they allowed free recombination here on the second chromosome. And second, we created females who are heterozygous for a second and third chromosome balancer, which allowed recombination only on the X chromosome. So 1 chromosome arm versus 2 chromosome arms. And we used changes in polymorphisms in order to identify sites of crossing over and gene conversion. And we sequenced individual progeny from multiple females from these crosses. So what did we find? Well, for crossing over, we found what we would expect based on Sturtevant and Beadle's data and based on others. Here I'm showing you data just for the second chromosome first. And you can see there's a subtle but clear overall increase in exchange in individuals from females who had multiple balancer chromosomes. So wild-type data here is in blue, and then data from from females carrying multiple balancers is in green. And what we see is an increase in centromere proximal and telomere proximal events. This increase is much more pronounced when we look at the X chromosome. So these are from females who had a balanced second and third chromosome. So there's a clear increase in centromere proximal and telomere proximal crossover events, and there's also a flattening of the best fit line. So because we whole genome sequenced individual flies, we could plot out individual crossover events. And I'm showing you all of the crossover events that we recovered here. I want to point out 3 things. The first is we recovered more double crossovers than we expected to by chance. This is in contrast to what you find in wild type, where you recover fewer double crossovers than you expect to by chance. We recovered 4 triple crossovers and 2 quadruple crossovers on the X chromosome. This is from 50 individuals, and then we got 1 triple crossover on the second. So in wild type, you get a triple crossover about 1 in every 1,000 myoses, and a quadruple crossover about 1 in every 10,000 myoses. So it was really cool actually to find these quadruples. I think I emailed Scott multiple times to tell him about that. And then second, for all these chromosomes which have 2 crossover events on them, all of these crossover events are widely spaced. And on average, it's about 9.5 megabases for our data. And that's essentially unchanged from wild type. That's telling us that interference is still acting on these chromosomes. These crossover events are still being widely spaced as they should be. So our question is, what's the mechanism for this increase in exchange? Are there more breaks? Or is there the same number of breaks, but you're repairing more of them as crossovers than you would normally? So in order to answer this, we have to find all the gene conversions. Finding the gene conversions on freely recombining chromosomes is actually quite easy because we did it for our initial whole genome sequencing experiment. And what we find is that while we expect to recover about 78 or 80 gene conversion events on those chromosomes that can undergo exchange, we actually only see 22. It's quite a drop. But the trick is finding the gene conversions on the balancer chromosomes. And we can only do this because we knew the structure and the sequence of these balancer chromosomes. And when we do that, we see a subtle but clear increase in the number of gene conversions on these chromosomes. So this is the second chromosome balancer up top and the third chromosome balancer on the bottom. So something's driving up the number of gene conversions on those chromosomes that cannot undergo exchange and bringing down the number of gene conversions on the chromosomes that cannot or that can undergo exchange. The other fun thing you can do with this data is you can plot out individual gene conversion events on the balancer chromosome. So here I'm showing you the second chromosome balancer, curly, and the third chromosome balancer, TM6, that we used. And you see that these conversion events are essentially evenly distributed along the genome. Not only that, they're falling almost right on top of the inversion breakpoints. And this is after one meiosis. So we find that 21 of the gene conversion events that we recovered were within 1 megabase of an inversion breakpoint. Now think back to the TM3 data that I showed you, where we didn't see any crossover events within about 2 megabases of an inversion breakpoint. So clearly, these conversion events are not responding to the interference that's generated by these inversion breakpoints. And I think the closest one we found was around 10 kb or so from an inversion breakpoint. So they're pretty much right on top of it. Okay, so now that we know the number of crossovers occurring in the genome and we know the number of gene conversions, we can back calculate the number of double-strand breaks. And that'll let us know if there's an increase in the number of double-strand breaks in response to the interchromosomal effect, or if the number of breaks is remaining the same. And when we do the math, what we find is that the number of breaks that are made in the interchromosomal effect is essentially identical to wild type, around 17 in both. And this is a very similar number to what you get if you look at breaks by cytology. So it's telling us that in response to multiple inversions, the number of double-strand breaks is remaining the same, but that double-strand breaks are being diverted from the gene conversion pathway to the crossover pathway. And those double-strand breaks that would have become crossovers on those multiply inverted chromosomes are actually being repaired as gene conversions. So the key takeaway here is that the recombination landscape is plastic in Drosophila, and that's not what we expected. In response to inversions, crossover numbers will go up on those chromosomes that can undergo exchange, and gene conversion numbers will go down. We also learned something really interesting, I think, about the timing of double-strand break repair in Drosophila. Originally, I would have told you that breaks are repaired as crossovers and gene conversions at the same time during meiosis. That's not what we're seeing. These breaks must be repaired at different times. You must repair breaks as crossovers first, and once you have the proper number of crossovers, you repair the rest of the breaks as gene conversions, because the cell is waiting. It says, I need to use these breaks, or I may need to use these additional breaks for something else, and that something else is a crossover. So of course it raised, these experiments brought up several additional interesting questions, I think, specifically about the placement of double-strand breaks. So we and others have spent a lot of time thinking about how are double-strand breaks distributed in Drosophila. We did spend a lot of time looking for motifs, similar to what we see in mammals, in some mammals where motifs will guide the placement of a double-strand break. We thought we've seen them in the past, but I don't think any of them have ever really panned out. This data makes me wonder, is it just random? Are breaks just made randomly in open chromatin in Drosophila, and the cell is just left with the problem of how to repair them? I think there's more work to be done, and it'll be interesting to do that. So where do we go from here? Well, fortunately, I don't think there's any shortage of ideas. I'm always one for more data, and I am actively working on getting half tetrads using compound chromosomes, so we can look at 2 of the 4 products of meiosis. I think Larry probably would have liked the idea of using compounds in experiments, from what I can tell from the literature. The other thing that I always love to mention is, Drosophila is a really, really interesting species group, and there's a lot of diversity over a relatively short amount of time. And I think it would be really fun to do similar experiments in closely related species or more distantly related species. See how the recombination landscape changes, and then use that as a guide to look for genes that might be influencing that change. And then finally, I've got to write grants at some point in my life, so I got to learn some buzzwords. And one of the buzzwords I'm working on now is machine learning or deep learning. So I think we've got a lot of outcomes of double-strand break repair, right? We've got gene conversions and we've got crossovers, and that's the kind of data that you can give to your favorite AI algorithm. and see what comes out on the other end. So it'd be fun to see if we can use that to determine what's guiding the placement of these breaks. So with that, I'd like to thank you for your attention. Thank you for the invitation. This was really fun talk to put together. I'm always looking for an excuse for a break from the hospital, and this was a great excuse for that. Specifically, though, I do want to thank Scott for all the support that he gave me during graduate school and for allowing me to join his lab. It was the best decision I ever made, that's for sure. I don't know about him, but Angie, my wife, who's here, who is responsible for everything that looks pretty and is spelled correctly in my presentations. Corey, she was a graduate student in the lab. She gave me some really great feedback on this talk, and then the core at Stowers was really supportive. Nicole Crown at Case Western, she was in Jeff Sokolsky's lab at UNC. We worked on the interchromosomal effect data in parallel, and she's been a really great collaborator and continues to be a really great collaborator. And the one thing I want to mention, I was really fortunate, I think, during graduate school to meet and work with a lot of really great folks on this project. Specifically, Dan Lindsley gave me a lot of help with the balancer work. And then John Merriam, who's here today, who actually made FM7, was a really great source of information while I was working on that.